The effect of AIP1 on the AB toxicity and longevity of Canorhabditis elegans by Jatin Savlani. In the world, over 6 million people are diagnosed with Parkinson's disease, a degenerative disease that causes confusion with nerve cells and affects daily locomotion. By the year 2050, over 40% of the human population will be affected by a neurodegenerative disease, according to the CDC. As seen in Figure 1, most of the budget for Parkinson's disease in 2010 was spent on research grants. Although this allows for further research on the disease, no attempt has been successful. Figure 2 shows the projected rate of Parkinson's disease up to 2050. As of now, 40.2% of people 65 and older that live in the United States will have Parkinson's disease. By 2030, 72.1% of all people that live in the US that are 65 and older will have Parkinson's disease. This number rises all the way up to 88.5% in 2050. Figure 3 shows the average amount of money that a person living in the United States that has Parkinson's disease has to pay each year for medical services. The most expensive cost is a nursing home, which costs around $5,000 per year, as many patients are not able to live by themselves. Next is medicine, which usually costs around $2,700 per year. C. elegans is a multicellular organism, which means that it goes through a complex developmental process, including embryogenesis, morphogenesis, and growth into an adult. Thus, biological information that we can learn from C. elegans may be directly correlated to more complex organisms, such as humans, which are somewhat more refractory to laboratory scrutiny. Figure 4 shows the general anatomy of a male C. elegans. Figure 5 shows each stage of growth that C. elegans take after an egg is laid. This process usually takes around 2 to 3 days. C. elegans have two naturally occurring sexes, a male and a self-fertilizing hermaphrodite. Females do not naturally occur. The majority of individuals are hermaphrodites. Males usually comprise of no more than 0.20% of the natural population. The number of males can be increased, however, by raising the temperature at the onset of sexual maturity. Hermaphrodites are protandrous. The individuals produce sperm first and then produce eggs. Most commonly, worms will simply fertilize their own eggs. However, the males that do exist copulate with hermaphrodites, thus mixing up the gene pool in the population. Eggs are laid within 2 to 3 hours of fertilization and hatch approximately 12 hours later. The worms develop into adults in 4 larval stages. This generally takes about 2 to 3 days when the temperature ranges from 20 to 25 degrees Celsius. Figure 6 shows the anatomy of a hermaphrodite of C. elegans, as Figure 7 shows another picture of the anatomy of a male C. elegans. In the article by Heather R. Brignall, she used C. elegans as model organisms for research on Parkinson's disease. Using fluorescently tagged polyglutamine repeats of different lengths, she examined the proteotoxicity of the C. elegans. Her results showed that the proteotoxicity of the C. elegans depended solely on the length of the polyglutamine repeat. Another characteristic she noticed was that the longer the length of the repeat, the shorter the lifespan of the C. elegans. Also, the ability to combine visualization of polyglutamine and live C. elegans with mutations with longevity allowed her to realize that increasing lifespan delays polyglutamine toxicity. This has direct correlation with Parkinson's disease in the way that Parkinson's disease speeds up polyglutamine and proteotoxicity. In the article C. elegans as a model organism to investigate molecular pathways involved with Parkinson's disease by Adam Harrington, he discusses that the accumulation and cellular stress induced by misfolded or aggregated proteins may underlie the neurodegenerative mechanisms leading to Parkinson's disease. Also, he says that in order to evaluate the effect of ASIN toxicity on neuronal activity and longevity, changes in worm behavior have been examined as a functional readout. His results showed that the more ASIN toxicity in each worm, the shorter the lifespan would be. Also, more genes were expressed when the toxicity was higher. In the experiment AIP1 AB amyloid peptide toxicity and C. elegans Parkinson's disease model by Wael M. Hassan, he tested the peptide toxicity in C. elegans as well as certain gene expressions. He also extended this analysis by examining earlier time points and by using a control strain that employed an identical transgenic construct to inductively express GFP in body wall muscle. Synchronous worm populations were grown at 16 to 32 degrees Celsius to the third larval stage and then were transferred to 25 degrees Celsius. To explore the possible applicability of their data to human disease, they examined whether human homologs of AIP1 could mimic the protective effect of AIP1 in their worm model. There are two human and mammalian homologs to AIP1, which are AIRAP and AIRAPL. The amino acid identities of AIRAP and AIRAPL to AIP1 are 43% and 38%. They found that AIRAPL but not AIRAP had a protective effect that delayed the paralysis phenotype of AB42 expressing animals in a manner similar to AIP1 protection, which is shown in Figure 10. In the article, SKN1 Dependent and Independent Regulation of AIP1 Expression Following Metabolic Stress by A. Ferguson, she tests for toxicity in C. elegans. Figure 12 shows Ferguson's methodology. Ferguson also followed the AIRAP amino acid to see if her data could correlate with humans. She had very similar results compared to Hazan, stating that the AIRAP did not have a protective effect. The purpose of this experiment is to have further research on Parkinson's disease and hopefully contribute to the cure. The hypothesis of the study is that the longer the length of the polyglutamine repeat, the easier the AIP1 gene will be expressed. First, I will separate my three strains of C. elegans: strain CL2179, strain 4001, and strain 4176, which is my control. Then I will synchronize the C. elegans using the synchronization assay, which is a 20% alkaline hypochloric acid to synchronize the C. elegans. Then I will apply the polyglutamine repeats, and then take pictures by photobleaching to show the AIP1 gene expression. This is my budget. This is my bibliography.